Hello guys, welcome to my YouTube channel. So today we are going to discuss Mendel's monohybrid cross. So what is Mendel's monohybrid cross? So first we have to know what does the term monohybrid represent. So we can break this term into two, mono and hybrid. So mono means one and hybrid means it is a combination of two things, right? So two different things. So monohybrid means offsprings. Okay, so these are those offsprings which differ in a particular gene, or as we say, Mendel used the term factor for it. So offsprings differing only in one factor. Okay, differing only in one factor. So if there is like a difference in two factors or genes, okay, between the offsprings, then we call such type of a cross as dihybrid cross. Okay, so here we are going to talk about a cross which will yield offsprings having difference in only one factor or one gene. Okay, so those kinds of cross are called as a monohybrid cross. So we will start with we will take two parents. Okay, P1 here means parents. We will take two parents. One parent will be having the phenotype tall and the other parent will be having the phenotype dwarf. Okay, and the respective genotypes for those parents are capital D and small d. Okay, so this is some sort of a representation. Some books or some authors use the representation of capital T for tall, homozygous tall and tallness, and small t for homozygous dwarf parents. Okay, so it is up to you whichever representation you want to use. You will proceed the cross with that one. Okay, so here we are using capital D as the genotypes. Okay, as a representation of the genotypes of tall parent, homozygous tall parent, and small d as the representation, genotypic representation for homozygous recessive or the dwarf parent. Okay, now we will cross it. So what will happen? It will lead to, first we will cross. That means we are actually making them we are actually making them sexually compatible. So the gamete formation, because you know gamete formation occurs through meiosis. Okay, so that means what? The chromosome number will get halved. So capital D will produce only one capital D, right, because of meiosis, and small d parent will produce one only small d because of meiosis. Okay, so we have the gametes here. Now what will happen when these gametes unite? When these gametes unite, you will get the F1 generation. So this is the F1 generation, where the genotype of the F1 generation will be capital D small d. Okay, and the phenotype will be tall. Why? Because capital D is dominant over small d. Okay, capital D is dominant over small d, right? So capital D is the factor for tallness and small d is the factor for dwarfness, and by definition tallness is dominant over dwarfness. So in this F1 progeny, okay, so this is the progeny that you get after first cross. Okay, so this is the progeny that you get. So in this progeny, it is having the genotype capital D and small d. So capital D will get dominated over, so capital D will dominate over small d, and the overall phenotype will be tall. Okay, tallness will be the phenotype. So this is the F1 generation. Now what will happen if you perform a cross between two F1 progeny? That means you will have two progenies, okay, having same sort of genotypic representation, capital D small d and capital D small d, that is both are hybrids. Okay, so these are all hybrids. And what sort of hybrids are they? These are monohybrids. Okay, so this is what I was talking about. So these are actually monohybrids. See, they differ in one particular factor. That is, one factor that they have in them is the factor for tallness, and the other factor that they have in them is the factor for dwarfness. Okay, so one factor capital D for tallness and one factor is for dwarfness. So they differ in only one factor, so they are called as monohybrids. So if you have a cross where you perform so if you perform a cross between two monohybrids, between two F1 progenies, what will happen? So by meiosis, obviously the monohybrids will produce either capital D or small d. Okay, not both. Okay, this here the graph, the picture shows capital D and small d are getting formed from this parent, but it is not, don't misunderstand, it is not like they will be formed simultaneously. So either this one will be formed, okay, or the small d will be formed, not both. It cannot happen that they will be formed simultaneously, because it's meiosis, right? The chromosome number needs to get halved. So either the first one or the second one. Similarly for this parent, either the capital D will be formed or the small d will be formed. Okay, so here we are talking everything in terms of probability. So what will happen? You have the four different gametes that could be formed. So in the F2 generation, this capital D can unite, okay, with this capital D, right? So if the capital D and capital D unite, you will get the genotype of the progeny as capital D, and the phenotype will be small, tall, and it will be homozygous tall because both the alleles are same, right? Now another possibility could be like that this capital D could unite with this small d, right? Then what will you get? You will get this one. The progeny will have a genotype of capital D small d, but the phenotype will be tall again because capital D is dominated over small, the capital D is dominant over small d, but it will be heterozygous because you will have two different factors within it, right? It is not the same factor. So another possibility could be like, so I will use a different color here. So what could be the another possible factor? So this small d could unite with this capital D. So in that case, you will again get a progeny with the genotype capital D small d, and phenotype will be tall because capital D is dominant over small d, and it will be heterozygous because two different alleles are there. And this small d could unite with this small d also. So it will be the genotype of such a progeny will be small d, and it will be dwarf because both are recessive alleles, and it will be homozygous, right? So these are all the possible genotypes and phenotypes of the progeny if you perform a cross between two different F1 monohybrids, right? So these are all the possibilities. Now don't think that all the four possibilities will arise simultaneously. So it is a misconception that all the four possibilities cannot arise simultaneously. So these are possibilities, okay, not like everything these progenies will, all the four progenies will be formed simultaneously. It cannot happen. So these are possibilities, and either one or either one of them can happen, right? Or both, or more than one also can happen, but it is not necessary that all the four will happen. Four progenies, four F2 progenies will be formed simultaneously. It is not necessary, right? So if you have a cross, so what the question comes is, they will give you a genotype. Say they ask you about a monohybrid cross, so they will give you a genotype of the parents. Okay, so you will get the genotype of the parents, you will get the phenotype of the parents. Okay, so they will ask you what will be the genotypes and phenotypes of the F2 generation. Okay, so here we perform for tallness. They can give from for some other phenotype, maybe some say short hair and long hair, or maybe someone with who is obese and who is not obese. So the phenotypes could differ, and accordingly the genes, the genotypes, and the representation of the alleles could also differ. But the main concept is the same, that when you have a parent, you have the genotypes. So by meiosis, the chromosome number will get halved. Okay, and then in the F1 you will have a monohybrid progeny, and when you perform a cross between two same monohybrid F1 monohybrids, okay, you will get 4 different possibilities in F2 monohybrids. Okay, now it is very important one thing. So what from here? Okay, so we missed one thing. So people will ask, what is the phenotypic and genotypic ratio? Okay, phenotype means which is visible from outside. Okay, so what is the phenotypic ratio for this? Okay, so phenotypic ratio means how many of them are tall and how many of them are dwarf. Okay, that is what we are dealing with a phenotype. So you can see here 3 phenotypes which have been tall, okay, and 1 phenotype which is dwarf, right? So the ratio for the phenotypic ratio is 3:1, okay, for monohybrid cross, 3:1. But what is the genotypic ratio? If someone says, so the genotypic ratio will be different. Why? Because the hybrids, the hybrids that you generated in F2, they show the same phenotype as the homozygous ones. Okay, they show the same phenotype, that is they are tall, but the genotypes are different, the genetic constitution is different, right? So in homozygous one you have only same type of alleles, that is capital D, capital D. But in heterozygous ones you have 2 different alleles, capital D and small d, right? So the genetic constitution is different. So you have 2 heterozygous, right? 2 heterozygous genotypes, one homozygous genotype for tallness and one homozygous genotype for dwarfness. So the ratio is 1:2, okay, is to 1. Why is it so? 1:2:1. So this one means the homozygous tall one. Okay, I am writing it as HT. So this 2 means heterozygous tall one. Okay, I am writing it as HTT, because you see in this F2 progeny we have 2 heterozygous ones, and this again one means the homozygous dwarf. Okay, I am writing it as HT. So this is also very important. So people will give you some problems, basic problems, where they will ask you, you have this number of total generations, F2 generations, calculate how many will be phenotypically tall and how many of them will be phenotypically dwarf. So suppose you have 16, okay, generations, someone says 16 F2 progenies. So out of this tall, so phenotype persons having phenotypically tall will be 3/4 into 16. Okay, so it is actually 12, right? So these are phenotypically tall, and phenotypically dwarf will be the rest, that is 1/4 into 16. So problems could be like this one. Okay, or someone will say you have 16 in total F2 generations. Okay, calculate how many are homozygous tall, how many are heterozygous tall, how many are homozygous dwarf. So then if you have to calculate genotypically, so then what will happen? HT, homozygous tall, will be 1 by, right? So 1/4 and into 16. Okay, so like heterozygous tall will be 2/4 into 16 and heterozygous dwarf will be 1/4 into 16. So like this you will calculate. Okay, so this is the importance of this problem. We will discuss more such problems in our subsequent lectures.